Now we're going to focus on some other characteristics of bacteria. And these characteristics are going to be related to some of the DNA technologies that we're about ready to get into. Remember we said that bacteria reproduce by binary fission, and that leads to no genetic variation because the daughter cells should be identical to the parent cells. But the thing is, we have lots of different types of bacteria, and they show lots of variation. So there are some processes that will lead to genetic variation in bacteria cells. So we're going to look at how bacteria cells can acquire new genes, which will give them genetic variation. Bacteria can acquire new genes through what's called horizontal gene transfer. So think of the horizon. The horizon is where the sky meets the ground. So the horizon is like that. The reason we call this horizontal gene transfer is because I can have one bacteria cell right here, and it can actually pass DNA to another bacteria cell sitting right next to it. So that is what we mean by horizontal gene transfer. Advanced organisms like us cannot do this. Imagine if we could do that. Then if someone has great hair, then they could just pass me the genes that code for their fantastic hair, and then I would have that characteristic as well. So obviously we cannot do horizontal gene transfer. In contrast, vertical gene transfer occurs when genes are passed from parent to offspring. So bacteria cells, they do both. They have vertical gene transfer whenever the parent bacteria splits into two offspring, but they also can have horizontal. So two members of the same generation can pass genetic information from one to the other. Horizontal gene transfer can happen in three ways, and you need to know them. There's transformation, conjugation, and transduction. We will go through each, and I will give you a way to help you remember the difference between transduction and transformation, since they sound alike and we get those confused. So basically what we're going to look at is how bacteria cells, like this one right here, is picking up new pieces of DNA. Once they pick up these extra pieces of DNA, two things can happen. These newly acquired pieces of DNA, they can be inserted into a plasmid. Remember, plasmids are extra circular pieces of DNA that we find in bacteria, or it can be inserted into the chromosome of a bacteria. Bacteria cells have one chromosome. And this diagram is trying to show you that. So it's showing you this gene could be inserted into the plasmid here, or it could be inserted into the chromosome, and that can give this bacteria cell a new trait. Now we see that this acquired DNA often has genes on it for antibiotic resistance. So now this bacteria cell is resistant to a specific antibiotic. So let's say penicillin will no longer kill it because it acquired these extra pieces of DNA. Now I wanted to show you a diagram that shows you exactly how bacteria cells can become resistant to antibiotics. Now notice I'm using the term resistant. Bacteria cells do not have immune systems. So never say that a bacteria cell will develop an immunity to. It cannot. It does not have white blood cells that travel around in the bloodstream and can fight infection. As a matter of fact, our white blood cells are like 100 times the size of one bacteria cell. But we say that bacteria cells or bacteria populations can develop a resistance to an antibiotic, never an immunity. So let's look at this diagram down here. So we have a plasmid, and we can tell that that plasmid has genes on it, and those genes could have been acquired through horizontal gene transfer. Again, we're going to talk more about those three methods in a second. And these genes are now coding for proteins that can affect antibiotics. So let's take a look what can happen. So here we treat a bacteria with antibiotics. The antibiotics goes into the cell, and now the cell makes a transport protein. So this gene codes for a transport protein that is embedded in the plasma membrane, and it can transport that antibiotic right out. So it does not kill the cell. We have a gene here that is coding for an enzyme that can break up the antibiotic. So obviously it's not going to kill the cell. And we also have genes that code for proteins or enzymes that can alter the antibiotic. So the antibiotic makes it in, but we're altering it and it's no longer going to affect the cell. Now, one thing you need to understand about bacteria cells and their plasmids is that whenever a bacteria cell reproduces by binary fission, it can make copies of both the plasmids and the bacterial chromosome. So we can see that the offspring has the same plasmids as this original cell. So if this cell survives and it divides by binary fission, pretty soon we're going to have two cells that have these antibiotic resistant genes in them. Now we're going to get into those three processes that we just wrote down: transformation, conjugation, and transduction. I'm showing you two different diagrams of transformation. We know that bacteria cells can die and they can disintegrate, and their DNA can be broken up into pieces, and now the DNA is floating around in the environment of other cells that are still living. When these cells uptake free DNA from the environment, then this is what is called transformation. So this diagram is showing you that there is DNA in the environment. There's free DNA in the environment, and some of those segments can cross through the plasma membrane of a bacteria cell. And you can see here that some segments can be incorporated into the chromosome, which is what you're seeing here. So now this cell is transformed, or we say transformation has taken place. Now you guys are actually going to cause transformation to take place in bacteria cells in the PGLO lab that we're going to do. What we're going to do is we're going to grow some bacteria, and we're going to put them in a little vial, like a little test tube, and we're going to add some extracellular DNA. We're going to add some plasmids to that same vial. Now I'm going to get into specifics later on about the PGLO lab, but something about this plasmid that we're going to mix in there with the bacteria cell is these plasmids have a gene that was placed on them by this company called Bio-Rad. And this gene codes for a glow-in-the-dark protein. Now this gene came from a jellyfish. So if the bacteria take up the DNA, or if they're transformed, then they should be able to make this glow-in-the-dark protein. And so the bacteria colonies are going to glow. Another gene that was put on this plasmid is a gene that gives them antibiotic resistance. Specifically, if they take up this plasmid, then an antibiotic called ampicillin will not be able to kill them. So we have to have good handling techniques, because we know that cells, bacteria cells, can take up free DNA in the environment. So we want to make sure that we're not exposing cells on your lab tables to this plasmid, because they can take that up, and we could create potentially dangerous bacteria cells in our high school lab that we cannot kill with an antibiotic that we used to be able to kill them with. So basically, you're going to transform bacteria. You're going to help bacteria take in these pieces of DNA. To do that, we're going to do two things. We're going to add a chemical called calcium chloride. And this causes bacteria cell membranes to become more permeable. We want to open them up so those pieces of DNA can get into the cytoplasm. And then we're also going to use a method which is called heat shock. We're going to heat shock this vial of bacteria and plasmids, and that's going to increase the chances that they're going to take up this extracellular plasmid that was generated by Bio-Rad. From there, we're going to grow these bacteria on agar plates, and only the bacteria that have the plasmid are going to be able to grow, because I'm going to put some antibiotics in that agar plate. So it would kill off the ones that don't have the plasmid. So the ones that have the plasmid are going to grow, and they can make that GFP protein, so they're going to glow. But all the bacteria cells that got put on those agar plates that do not have the plasmid, they're going to die. So we'll be able to select and grow just the cells that contain that DNA that we introduced into their environment. Let's look at that second type of horizontal gene transfer, which is called conjugation. If bacteria are conjugating, then basically they're hooking up and exchanging genetic material. One bacteria can transfer DNA from one to the other via the structure that you see here, which is sometimes called the sex pilus. So you can see the bacteria has to have cell-to-cell contact. We can see that the DNA is being replicated. Remember, DNA polymerase, that's the enzyme that is making a polymer of DNA. So in this situation, the plasmid is being copied, so there's two plasmids, and it's being transferred to this cell, the recipient. In a way, this is kind of like sex in bacteria cells. But it's not leading to new offspring, it's just leading to the ability to horizontally pass genes. The third type of horizontal gene transfer is called transduction. And it took me forever to find a picture of a ducking duck. To help you remember that transduction has to do with viruses. Think of duck, a virus is coming. Take cover. So transduction involves the transfer of DNA from one bacteria to another via a virus. Remember, a virus that can infect bacteria cells are called bacteriophages. So let me show you with this diagram how a virus might take DNA from one bacteria cell and transfer it to another one. Now, the color coding of this diagram is necessary. Here we have virus DNA or RNA. So here's viral DNA. And we can see that it gets injected into this bacteria cell. So now we have virus DNA, purple, and we have the bacteria genome that can get digested up into small bits. The bacteria cell is going to be producing new viruses, and you can see right here, this green part, this is some bacterial DNA. And it got packaged into this virus. So this virus contains viral DNA and it also contains bacterial DNA. When this virus infects a new bacteria cell, then it can inject both viral DNA that we see here in purple, but also it can inject this DNA from a different bacteria cell. So now we have horizontal gene transfer, and this bacteria cell now has new genes. Now remember, after I finished talking about protein synthesis and gene regulation of eukaryotic cells, I stopped and I taught you about the reproduction of viruses called retroviruses, like HIV. At that point, you had a good understanding of protein synthesis, so I thought it would be easier for you to understand virus reproduction at that point. Now I'm going to talk about two different cycles to virus reproduction: the lytic and the lysogenic cycles. Basically, viruses can enter cells and they can immediately become active. That means the cell is going to immediately start producing new viruses. Or that virus can be dormant and that cell will not produce new viruses immediately. The fact that viruses can do this, they can enter the lytic cycle or the lysogenic cycle, is well understood in prokaryotic cells. So that's why whenever we teach you about the lytic or the lysogenic cycle, we usually do that as we're talking about bacteria cells. All the diagrams that you're going to find, if you Google lytic cycle or lysogenic cycle, you're going to see that we have bacteria cells and they're being infected by bacteriophages or viruses that can infect bacteria cells. So again, all these diagrams that you see are always going to be about bacteriophages and how bacteriophages can show the lytic cycle or the lysogenic cycle. But again, there's other viruses that can affect eukaryotic cells, so that can either go into the lytic cycle or the lysogenic cycle. To help you remember what takes place in the lytic cycle, I like to think of lytic is cryptic. A crypt is a place where you put a dead body. So crypt or cryptic means death. If a virus enters the lytic cycle, then that's going to lead to immediate destruction of that infected cell soon after infection. So here's a diagram of the lytic cycle. So we have genetic information being injected into the bacteria cell. We can see that this cell is immediately starting to make new viruses using that viral genetic information. And then we see that the cell is going to die whenever the viruses burst through the cell membrane. So if this is what happens to a bacteria cell after infection by a bacteriophage, then we say that that virus was in the lytic cycle. Let's compare that with the lysogenic cycle. So again, a bacteriophage is injecting its genetic information into a bacteria cell. And we can see that the genetic information of the virus becomes incorporated into the genome and it stays there. And we can see that the cell is not immediately producing new viruses. And the cell is dividing. So when the cell divides, it copies its DNA, including the viral genetic information that was inserted into the chromosome. And now we have two cells that are ultimately infected by this virus. So we say that the virus is in a dormant stage of reproduction. It's entered the lysogenic cycle. Now, this virus can go from being in the lysogenic cycle to going into the lytic cycle. And that's what this part of the diagram is attempting to show you, is that there has been a change in the environment. And that triggers this virus to become active. So now the cell starts to make these virus components, and eventually that cell is going to die. Now these viruses that are in the lysogenic cycle, they will stay dormant if they are living in an environment where there's plenty of resources. So basically life is good and there's no reason to move out of that bacteria cell. However, if there's a change in the environment, then that's going to trigger these viruses to enter this lytic cycle of reproduction. And basically, they're leaving a cell that's in an environment in which resources are not plentiful, and it's going to maybe move to a new environment where there's plenty of resources. Now, let me relate the lytic cycle and the lysogenic cycle to a virus that infects eukaryotic cells. So cold sores are caused by the herpes virus. And people who are infected with this herpes virus, they don't have cold sores all the time. So the virus is in those cells and it's dormant. But if conditions change, then that virus can become active. And then you start to see a cold sore start to develop as those cells that are all infected with that virus are being destroyed, as more viruses are being made and released from those cells. So at that time, then that herpes virus can be spread from one person to the next. So you shouldn't be kissing on anyone that has cold sores, because that means the virus is active. It has entered the lytic phase and it's spreading, or it can spread. Now think about what I said though. Changes in that person can cause that virus to become active or enter that lytic stage. So when a person starts to get sick, whenever a person becomes stressed out, then that's whenever they start to develop cold sores. So that is the change in that environment or in that person that is triggering these cells to become active. I included this diagram down here just to emphasize that in the lysogenic cycle, these cells are going to divide and you're going to end up with many infected cells with dormant viruses in them. That diagram above really didn't show you that you get a bunch of these cells that are infected and have these viruses that can become active. Another reason why I included this diagram is I wanted you to see this term prophage. When I'm saying prophage, I'm basically saying virus DNA. It's the DNA that is going to code for the proteins and the new genetic material of those new viruses. Now, I don't want this next diagram to confuse you with transduction. Remember, transduction is when viruses can introduce new DNA into bacteria cells. But I want to show you a way that viruses can get genetic variation. If you have two viruses, so here's two viruses, and let's say that they're different types of viruses, but they're very similar. If they infect the same bacteria cell, so here's the genetic material of one, here's the genetic material of the other, then whenever these new viruses are assembled, then we can create viruses that have a genetic combination from both of these parent viruses. So now this progeny, this offspring, has new traits. So again, just like bacteria, viruses can pick up different combinations of bacteria in different ways, leading to genetic variation. I believe oftentimes students get viruses and bacteria mixed up, and they pretty much think they're the exact same type of organism because they both can cause disease and infection. But I want to take a second after we've studied both ideas about bacteria and viruses to compare the two so that you don't mix up some of this information that we just talked about. So starting with bacteria, remember bacteria are living cells. They do not have a nucleus, they do have DNA and ribosomes. Viruses are not considered living. They have a protein capsid and they have genetic information, either DNA or RNA. When bacteria cells need to produce their proteins, they have the machinery to do it. They have their own enzymes like RNA polymerase, they have their own ribosomes, and they can go through transcription and translation. When viruses need to reproduce and make new viral proteins, they have to use the machinery of the host cell. When bacteria cells reproduce, that is by binary fission. When viruses reproduce, it's either going to be through the lytic cycle or the lysogenic cycle. We said that bacteria can gain variation, and I'm adding one in here that I hadn't talked about, but we've talked about it in previous lectures. Bacteria cells can gain mutations. They can gain new genes through transformation, which is taking up of extracellular DNA into a cell. Conjugation, which is transferring through a sex pilus. Transduction is through virus infection of bacteria cells. And viruses can gain variation as well. They can gain mutations. Remember, especially RNA viruses are more apt to mutations in their DNA. And also, if two similar viruses that we just talked about infect the same cell, then it can allow recombination of the viral genome and new traits in the new viruses that are produced. I indicated earlier that we have to teach you some of these ideas about bacteria cells because we're going to relate bacteria cells to some of the DNA technologies that we can utilize. We've already talked about copying DNA or amplifying DNA, and we did that in the last unit after we talked about DNA replication. I taught you about the technique called PCR. So that is one technology that we can use to amplify DNA. But now I'm going to get into how we can use bacteria to clone DNA or amplify DNA. Now, there's 4 processes to understand if we're trying to get bacteria cells to copy a segment of DNA for us so that we can do research on it. First, you have to understand how we can take this DNA to be copied or amplified, how we can get it into a plasmid, or how we can engineer the plasmid. After we've engineered a plasmid, this is now called recombinant DNA. Recombinant DNA is whenever you have DNA and it is made of DNA from 2 different organisms. So in a plasmid, we can take bacteria DNA in the plasmid and we can actually mix it from DNA from humans. We can insert a human gene that codes for a protein. So that's an example of making recombinant DNA. The next process to understand is how we can transform the bacteria. We've already talked about transformation, and we're going to do this in the Pico lab. We're going to heat shock. That is one transformation method, and we're also going to use a chemical calcium chloride that makes the bacteria cell membrane more permeable. Another idea to understand is after we have exposed bacteria to this recombinant DNA to try to transform them, how can we select for just the bacteria that was transformed? And we can get rid of the other bacteria that do not acquire this recombinant DNA. And then eventually we're also going to talk about, well, how can we grow these cells with recombinant DNA and then remove the DNA and purify it so that it can be used in a lab. This diagram summarizes some of those different processes. We can make recombinant DNA, add it with some bacteria. We can transform that bacteria by heat shock and calcium chloride added to the test tube. And then we can select for the transformed bacteria. If we grow them on a plate that has antibiotics on it, and the plasmid has a gene coding for antibiotic resistance, then they'll be able to survive. So we just selected for the transformed bacteria. And this plate would basically be our control plate to compare the other plate to. On this page, I'm going to start to go through the specifics of these 4 big ideas that you need to understand to allow bacteria to clone DNA for us. So the first thing that we're going to look at is how we can engineer plasmids. When we have plasmids, and they're a combination of DNA from 2 different species, we call them recombinant plasmids or recombinant DNA. To abbreviate that, you just put an R there in front of DNA. Now, because the structure of DNA is the same for all organisms on Earth, and the flow of information from DNA to RNA to protein synthesis, which is the central dogma, since it's the same for all organisms, then that is evidence of a common ancestor. Another way to say that it's common to all organisms is we say that DNA structure and the central dogma is universal. That means all organisms show this. Now, the fact that the flow of information is universal, that means that we are able to take DNA from other species, put it into the cell of a different species, and that cell will be able to produce the protein that that gene codes for. So first, let's talk about how we can make this recombinant DNA. We need to take a plasmid, and we need to open it up so we can insert some DNA. So we have to cut it. Now, a tool that we can use to cut DNA is called a restriction enzyme. Here's an example of a restriction enzyme. Now, we're going to cut the plasmid with this restriction enzyme, but we have to cut out the gene from the other organism with the same restriction enzyme. That's very important. From there, all we have to do is we have to mix the 2. So we have the open plasmid, we have this segment of DNA from a different organism. We have to mix the 2, and we're going to add a sealer. Remember, ligase sealed the backbones during DNA replication. Well, ligase is going to seal those spots where the 2 different types of DNA join together. So we're going to add ligase, and it's going to seal the backbone, and we now have recombinant DNA. It's a piece of cake. Now, here's some more information about restriction enzymes. As I mentioned, they're an important tool used in different areas of genetic research. Restriction enzymes were first discovered in bacteria cells. So the name of the restriction enzyme is after the bacteria cell in which they were discovered. So that first one that we indicated on this diagram up here, EcoR1, that's a restriction enzyme that was found in a strain of E. coli. Here's some names of restriction enzymes. I'm just going to put RE for the restriction enzymes. So those are the abbreviations we use. And this is telling you the bacteria strain that we isolated them from. And then over here, this is the DNA sequence that these restriction enzymes will bind to and will cut. So each restriction enzyme binds to a specific sequence. And that specific sequence is called the restriction site. So for this restriction enzyme that you see down here, we say this area is called the restriction site. Now, when the DNA is cut by the restriction enzyme, then you're going to end up with some base pairs that are not matched up. So for example, when this restriction enzyme cuts here, so it recognizes A, G, C, T, T, and whenever It sees that sequence, it can bind to it, and it's going to cut right here. So it cuts between those two A's. And if we open up the DNA here, you're going to see this A is no longer bound to a T. This G is no longer bound to a C. Same goes for the DNA on this strand. That T is not bound to an A. We cut them apart. So we say that these ends that are generated by the restriction enzymes are called sticky ends. These sticky ends have unpaired bases that we can use to join other DNA in that area. As I mentioned, you have to use the same restriction enzyme to cut open the plasmid and to cut out the gene of interest that you want to insert into the plasmid. So notice that the plasmid and this gene of interest, or the DNA from the different species, they have to have the same restriction sites for a certain restriction enzyme. If you use the same restriction enzyme, then these ends are going to be complementary to each other. And the reason why this DNA will join with this one is because G will pair with the C, and A will bind to the T, and T is complementary to the A. So this fragment of interest is going to join to, or anneal to, the sticky ends of this plasmid. So after we seal up the backbones with ligase, we now have recombinant DNA. So that plasmid is referred to as recombinant DNA. So what can we do with recombinant DNA? Because we're going to get some from a lab and we're going to insert it into a bacteria cell to prove that it's pretty easy to insert foreign DNA into bacteria cells or transform them. So we can take recombinant DNA, we're going to put it in a bacteria cell. And the reason why we would want to do this is because we can make this bacteria cell produce a protein. An example is human insulin. Before this technique, we would get insulin from pigs and cows because their insulin protein is very similar to our insulin protein and can activate the same signal transduction pathways as human-produced insulin. Again, evidence of a common ancestor when you can take molecules from another organism and utilize them in a different organism. I digress. Now what we've done is we have made recombinant DNA. So we have made plasmids that contain the gene for insulin. We place them in bacteria cells, and then the bacteria cells all day long transcribe and translate that gene, and then we can isolate and purify the insulin. Another reason why we would insert plasmids into bacteria is to finish what we started with. Remember, we were trying to amplify DNA. We want to copy it. We need to make a lot of copies of it or amplify it. We can put it into the bacteria cells, and the bacteria cells are going to reproduce by binary fission. So I will end up with millions of copies of DNA in 24 hours. Continuing on with this idea of what can we do with recombinant DNA. So again, we can use it to make a lot of protein. We can research that protein. We can use it to develop medicines. We can use this protein to help us make plants that are resistant to certain pests and insects. Can even engineer bacteria to clean up oil spills or other toxic spills because we're introducing genes into them that will allow them to produce enzymes to break up these molecules. Here we have many copies of a gene. Again, we can use it to do research on that gene, to develop medicines. It can benefit the food industry because we can do research on that gene to figure out how we can introduce genes into livestock to make them grow faster and produce more tissue for food. Now, I want to go through a process so you can understand how we can take recombinant DNA and we can use it to modify an organism. Whenever we modify an organism, they are called GMOs. And we're going to talk more about GMOs later on in a lecture. But I want to introduce some other technologies that we can use related to GMOs and gene therapy. So this process starts out with what we just talked about, making recombinant DNA. We can cut out the gene, and we're going to use restriction enzymes to do that. We cut open the plasmid, we add some ligase, we can make recombinant DNA. We We can take the recombinant DNA, we can transform the bacteria cells using heat shock, and now we have bacteria cells that will divide, and they will amplify the DNA, so we have a bunch of it. Now what we can do is we can take this DNA and we can coat gold particles with it, and we load it into a special type of gun. This gun, this gene gun, is going to shoot these gold particles at some cells. Here I have some newly developed plant tissue, and we're going to shoot these particles at this plant tissue, and some of these cells are going to take up this specific gene. This gene right here that we first put into this plasmid. And that gene now can code for a new trait in those plant cells. Now, not all the cells are going to take up this DNA and be transformed. So we want to be able to select for just the cells that have these new DNA genes. So we're going to grow them on media that allows us to select for just those cells. Whenever those cells start to grow and divide and form little pieces of plant tissue that are viable, then we can transfer them and we can grow them in a different environment, and they can grow into a genetically modified plant that now has a different trait. Maybe it's resistant to insects. So that gene codes for a protein that the insects find distasteful, so they will not chew on the plant and kill the plant. Or we maybe introduced a gene that codes for herbicide resistance. So we can spray our garden and the weeds are going to die, but our tomato plants aren't, because that herbicide is not effective against them because of this new gene that they acquired. So let me just say, we've been genetically modifying plants for a long, long time. So all this hype about GMOs drives me nuts. If we have not genetically modified these crops that we're growing, then a tomato in the grocery store would be about as big as a grape, and it would be ridden with holes because insects would be chewing on it. So genetically modified organisms is allowing us to do a lot of things in agriculture that we would not be able to do. We can produce the amount of food that we produce today to feed billions of people because we have genetically modified organisms over time. Now I want to focus on this selection part. So we talked about how we can make recombinant DNA, how we can insert it. Now we're going to look at how we can select for the transformed cells. So remember, we're inserting a gene that codes for a specific trait into the plasmid. But you can see here that these plasmids are also going to have genes on them that code for some kind of resistance, like antibiotic resistance. So this gene on this plasmid, if it's in a cell, is going to allow that cell to have antibiotic resistance. Lots of times we also use genes that code for a protein that allows us to simply look at these organisms and identify that they're genetically modified. So this gene can code for like a blue protein. Or in the Pico lab, there is a green protein that this gene codes for. So we can look at these colonies, and if they glow green, then we know that they were genetically transformed. So let's talk about how we select for those transformed cells and we kill off the rest. When we go to transform our cells, our bacteria cells in the Pico lab, we're going to add the plasmid. So let's show the plasmid. In goes the plasmid. And again, the plasmid has already been engineered for us. It was engineered by a company called Biorad. They just ship it to us, and we're just going to put it in a vial. And then we're going to take some bacteria, and we're actually going to grow them. So you learn proper growing techniques of bacteria in the lab. And we're going to take these bacteria that we grow, and we're going to put them in here. Notice, they do not have this plasmid. They just have their single chromosome that all bacteria cells have. From here, we're going to put them on agar plates. So we're going to take a sample, and we're going to put it on this agar plate. We're going to take a sample from this vial as well, and we're going to put it on this agar plate. One of our agar plates is going to contain ampicillin. Remember, we've engineered this plasmid to contain a gene for AMP resistance. So this cell is going to survive, and it's going to grow into a colony of billions of cells in one little area. We'll see a spot growing, And that spot originated from one bacteria cell that was not killed, dividing into 2, into 4, and so on. But all these other ones that were not transformed, so let's indicate not transformed, because not all of them are going to be able to take up this plasmid. So this one's not transformed, same with this one. All of these cells are going to die. So basically, we are selecting for the cells that were transformed. So what we end up seeing is we're just going to see 3 spots where we had 3 cells that were transformed, growing into colonies. Now over here, our control plate is we basically have a plate of agar. It's got lots of food on it, just like this one had food on it as well. And this is our control plate, and we should see plenty of growth that we can use to compare to the growth that we see on this plate. So on this plate, we're going to get both transformed and non-transformed bacteria growing. So we're going to see lots of bacterial growth on those control plates. Now I want to take a second to show you problems related to restriction enzymes, plasmids, and restriction sites. These can be simple problems, but not if you haven't seen them before and practiced them before. So here I have a plasmid, and you can see that oftentimes they're going to indicate how long in base pairs this piece of DNA is. So this one is 5,400 base pairs long. So it's a pretty small segment of DNA. The first question says, how many segments are produced if the plasmid is treated with indel and Hind3, which are 2 different types of restriction enzymes. If you want, hit pause and see if you can figure out this answer before we go through it. Remember, restriction enzymes will recognize a restriction site, and the restriction sites are labeled for you here. So this is telling you that Hind is going to cut right here. So if we treat it with, let's do this first one, we'll call it indel. So here's restriction site. So this restriction enzyme is going to cut the plasmid here, here, here. And if we treat it with Hind, it's going to cut here, here, and that's it. So now we have to count up how many pieces of DNA would we have floating around after we treat this plasmid with restriction enzymes. Well, here's one piece right here. So here's one piece, here's another piece, 2 pieces, 3, 4, and all that would be 5. So your answer would be 5. Now I race to answer the next question because it's about that same plasmid. So example 2 says, identify how many segments are produced and the approximate length of each segment in base pairs if the plasmid was cut with Hind3. So we're going to treat it, and the restriction sites are here and here. Now if you think about it, this looks like about, from here to here, would be about 25% of the plasmid. And this segment ends up making up about 75% of the plasmid. So the answer to the first part of the question is we would end up with 2 segments of DNA. And to figure out the length and base pairs of this segment, we would have to take the total number of base pairs, times it by 75% or 0.75. And to figure out the length of this segment of DNA, it would be, again, take the whole and times it by 25%. This would give you about 4,050 base pairs long. So that's this segment. Let's indicate that. So this segment would be about 4050. And this segment would be about 1,350 base pairs long. So that would be this one right here. Let's look at a different type of problem now. Example 3 says, when plasmid B is cut with different restriction enzymes, the following fragment lengths are produced. This table is showing you that if we cut with this restriction enzyme, then we're going to end up with 2 pieces of DNA. If you cut with BAM, then you only end up with 1 long piece of DNA. And then if you put both of them in there with that DNA, we would end up with 3 different segments. So with this information, they want us to show on this plasmid where the different restriction sites are for both the ECO-R1 and the BAM restriction enzymes. They also want us to indicate the lengths and base pairs of each of the segments of the plasmids. First of all, you can tell from here and from here the length of our plasmid. Our plasmid is 1,000 base pairs long. First, I'm going to start with ECO. So it says that if I treat this plasmid with ECO R1, that it's going to cut in two places, and one segment will be 400 base pairs and the other one's going to be 600. So let's say that we cut here, and I'm just going to draw an imaginary line here that I'm going to erase here in a second. But if we cut here and the other restriction site was here, does that make sense that this segment would be 500 and this segment would be 500 base pairs long? So we know this is not right for this other restriction site. So I always like, again, to cut basically at 12 o'clock and then figure out where the other restriction site's going to be. And I believe it would probably be about, let's say, here. So this segment would be the one that's 600 base pairs long, and this segment would be 400 base pairs long. And we would want to label these restriction sites if it asks us to do that. And basically, we're just labeling it with the restriction enzyme. So this is a restriction site for ECO R1, and this is a restriction site right here as well for ECO R1. But we're not done yet because they want the restriction sites for ECO R1 and for BAM. So now we have to figure out where BAM cuts. And I can tell that BAM only cuts in one spot. There's only one restriction site. So I'm just going to add this information down here. So we know that this was a restriction site for ECO R1 and here as well. So put ECO R1. And let's figure out where BAM is cutting. Now you have to look at this third column to figure out where BAM is cutting at. If you notice, I have a segment here that is 400, and then these two segments, they add up to 600. So I bet the restriction site is somewhere on this segment that was 600 base pairs long. So if there's a restriction site here for BAM, then I should get, when treated with both restriction enzymes, I should get a segment that is 100, a segment that is 400, and a segment that is 500. Let me show you, though, that there can be different answers, both correct, for this problem. Because if you think about it, I cut with ECO here and here. Remember this segment right here, this one was 600. If I have a restriction site here for BAM, it would still give me the same results. So let's put ECO here, BAM here, and this would be 100, this would be 500, and then this one would be 400 base pairs long. So these types of questions, sometimes they can take you a while, but they're pretty easy to do. But if you have not practiced them, then you will not be able to finish them in the time constraints of the test. Now this next problem, make sure that you understand how to read a gel electrophoresis, and that's coming up in another lecture. So I'm introducing a little bit about gel electrophoresis here with this plasmid question. So what this gel is showing us is that if you cut DNA with ECO R1, then it's going to cut it into two segments. And this segment is 70, see this over here, is 70 kilo bases long, and this one is 30. So you're just using the gel electrophoresis to help you figure out how long these different segments are. This one would be 60, this one's 40 in length, and then when you treat the DNA with both restriction enzymes, you get a 40, a 30, a 20, and a 10. So this first part says, how many restriction sites were on the plasmid for the restriction enzyme ECO R1? So it's a plasmid. Make sure you pay attention to whether it's a plasmid or linear DNA, because you would get two different answers. So if you have a plasmid, then you would have to cut, here's one cut, and that would just give you one piece of DNA. It'd just be one long piece of DNA. There has to be a second restriction site. So the answer to this is there's two restriction sites, and that would give you one piece of DNA, and here's the other piece of DNA. Now, I want to show you if it was linear DNA, so if we were cutting like eukaryotic cell linear DNA, and you would only need one restriction site. If you cut here, you would get 2 pieces. Does that make sense? So cutting linear DNA is going to end up with different pieces than cutting circular DNA that has the same number of restriction sites. So let's go down here, and it says on the following plasmids, sketch the approximate locations of restriction sites for ECO R1. Indicate the length of each segment in kilobases. Again, hit pause if you want and try to answer the question and see if it matches what I'm going to get here. First, we should probably note that this plasmid is 100 kilobases, or kilobase pairs long. And I always like to cut at 12 o'clock. And think about if I cut at 12 o'clock and 6 o'clock, then those segments would be 50 base pairs long, or 50 kilobases long. But I need 70 and 30, so I'm going to cut about right here. So that means this segment would be 70 kilobase pairs, and this one would be 30. On this plasmid, it wants us to put all the restriction sites. So for ECO and for the other restriction enzyme, which is HAE. So again, let's put our restriction sites on there for ECO. We determined they were there. Got to label them. I'm not going to put 70 and 30 on there because I'm going to cut with the other one, with HAE. And going over here to this third column, we can see that when you cut with both of them, we're going to end up with 4 pieces. And right away, my brain is picking up that I could take this 30 segment. Remember this segment right here was 30? And I could split it into 20 and 10. And I can take the 70 and I can split it into 40 and 30. So let's do that first cut. Let's take 70 and split it into 40 and 30. Remember, the whole thing is 100. So splitting it at the 6th position would give you 50. So we need to go to 40. So I would say 40 is probably about here. So that means this segment is going to be 40 kilobase pairs long. This one would be 30. And now we have to cut the 30 into a 20 and a 10. So again, you can cut different places. I mean, I'm going to go ahead and just cut it here. And this is my segment that is 10. And then this one is 20. But I also could have cut it here, making that bigger piece 20 and this little piece right here the 10. But let me get rid of that so you're not confused. Now we just need to indicate that the restriction site for HAE was there and also here.